Welcome grade 12s. It is Life Sciences time with Looney and Aslam. We're here to help you with all the things you need to prepare for your exam revision. So Aslam, what are we doing for the grade 12s today?
Today, Looney, we are going through the entire paper 1. We are going to do revision for the Gauteng learners. That's a big blessing because they're writing paper 1 on Monday. We want feedback from all the others, Looney, to tell us. Talk to those guys on the Facebook. Tell them we want to know when they're writing in their different provinces.
All right. Guys, you heard Aslam. Tell us when you're writing and tell us if we're really helping you, because we're here for you guys, and ask us as many questions as you may have. We do have Curio for Life Sciences as well. The questions are up, guys. Answer as many as you can, and it will tell you whether you're right or wrong once you press the submit button, guys. So press the submit button, check if your answers are right. If they aren't right, guys, go over them, see what you've done wrong, and Aslam will help you right at the end of the show with the questions. We do have Facebook, guys, so go on to Facebook. It's facebook.com/learnextra. Our Learn Extra page, guys, where you can download the notes, the videos and the schedules. It's learnextra.co.za/live. Our handle on Twitter is @learnextra. So make sure you have all of that, guys, to communicate with me and talk to me on our page. Ask me as many questions as you may have, and Aslam will help you right now. Aslam?
Thanks, Looney. Welcome back, guys, and let's get started. I thought we'd put just one Section 8 type question up just to warm us up, and at the same time to introduce all of you to this type of question. Not introduce, rather to refine the way you answer this type of a question. Let's read it quickly. Indicate whether each of the statements in column 1, that's column 1, applies to A only, or B only, or both A and B, or none of them in column 2. So they're giving you something in column 1, and in column 2 they're giving you something else there, an explanation or whatever else, and you need to match it. So it's called multiple matching. And you notice this particular matching generally has 2 marks each, because you need to make a selection. You've given choices, you need to make a selection, and it gets more refined in that you've got to say whether it's A only, B only, and so on. So that's why you're getting 2 marks for that. So the first one says, 1.1, examples of a reproductive isolation mechanism. And column 2 says adaptation to different pollinators, flowers adapting to different pollinators, or breeding at different times. A reproductive isolating mechanism is something that's going to isolate the organism from reproducing or interbreeding. So obviously then in this case it would be A and B, both of them. Flowers that adapt to different pollinators, because of speciation, they will be reproductively isolated because different pollinators will come to them then. And other organisms breeding at different times of the year or different seasons, mating seasons may change, that will fall there. Fossils found in the Great Rift Valley. And that one there would only be A. If you are wise enough, then you'd know that town child, and you are from South Africa, you'd know that town is in South Africa and it's not in a Rift Valley. So therefore that one is out. It's Nutcracker Man, that's A only. Supports the out of Africa hypothesis, Y chromosome mutations, and migration of Homo erectus. We're going to talk much about that later, so we just go straight to A and B in this case. They both support the out of Africa hypothesis. 1.4, characteristics that may be used by scientists to build a phylogenetic tree. A phylogenetic tree, a tree to show relationships between organisms and ancestry. Similarity of DNA sequencing, fossil evidence. Do we look at only one of them? Do we look at both of them? Do we look at none of them? We will look at both. We look at the fossils, and we will look at the DNA sequencing and how similar or how different they are. Foramen magnum is towards the back of the skull. Human, chimpanzee, wow. Human A, chimpanzee B, and the answer there is straightforward, guys. It would be A only. Back of the skull. Sorry. There. See how easy I made it, Looney. I made it easy wrong. Okay, towards the back of the skull. That's what's so important. You must read. The back of the skull. If the foramen magnum is towards the back of the skull, it forces the vertebral column to come in a more horizontal way, and therefore making the organism quadruped instead of bipedal. So obviously it can't be A, it would be B only. Natural selection, Erasmus Darwin or Lamarck. There's a catch. You know very well that it can't be Lamarck, so you knock him out, but then that's Darwin. But notice the word that we use, Erasmus Darwin. So Erasmus Darwin did not propose the theory of natural selection. It was his grandson, Charles Darwin. So it is neither A nor B, none of them. Law of inheritance of modified characteristics, Lamarck or Wallace, that's a simple one, it's A only. And the epoch in which we are currently living. I felt it was interesting to put that one in because we just discussed that on Wednesday's show. And we're looking at the epoch, Holocene or Pleistocene. You have to know your work here, obviously. You can't take chances, it's not an understanding. You either know it or you don't know it. In this case, it is the Holocene. Remember, the one that we spoke about, Anthropocene, on Wednesday, that is a hypothesized. They're saying that we should be calling this the Anthropocene. But we are actually now, according to the scientists, they agree that this is the Holocene. So this is how you answer this question. Remember, you do not do it the way I've done it here. You're not going to do it on your question paper. You're going to write down 1.1 to 1.8, and next to each you're going to write the A only, or none, or both. That's how you answer this question. You do not have to rewrite the whole question. We find when we're marking, lots of kids rewrite the whole question, and then they're not following the instructions. You're going to lose marks because we didn't ask you to write these things down. We asked you to write one of these options only. Only those things. I'm going to just undo some of that because when you look at it later, you may think that's the answer, and that's not the answer. That was just an example. Good. Let's move on to some section B type of questions. Remember, we are, if you've noticed by now, we're dealing with evolution, and the following was taken from the prep of last year, Gauteng and Free State. The pictures below show views of skulls from Homo erectus and Homo sapiens. Study the diagrams and answer the questions. So they're giving you a skull set 1. There's a skull set 1 and a skull set 2. So we have, this is skull set 2, that's skull set 1. And the questions that follow, which skull set represents Homo erectus? Justify your choice by referring to 2 structural features. Which one of these 2 sets? Remember, each set belongs to one of them. Each whole set there, they're giving you the side view, the front view, and the bottom view in terms of what you're seeing there. So they want to know which of these 2 sets belongs to Homo erectus, and they want to know from you, why do you say that using structural evidence? And the answer is obviously skull set 2. Why? Because you have pronounced eyebrow ridge. It has prognathus. It is prognathus. It has a protruding jaw. Remember, if you do not use the word prognathus, you can talk about protruding lower jaw especially. It has a flatter skull, and it has a smaller brain capacity compared to the Homo sapiens here on top. Next question says, name one fossil site in South Africa where Australopithecus fossils were found. And the answer is either you say Kromdraai or the Sterkfontein area or Taung. Those are the 2 answers that we can give. The out of Africa hypothesis states that Homo sapiens originated in Africa and spread from here to the rest of the world. Good. I like the way the question is phrased because that's already telling you what the out of Africa hypothesis is. Sometimes the examiners ask you, explain the out of Africa hypothesis. Now this question is telling you what it is. What is it? It says that the Homo sapiens originated in Africa and spread from here to the rest of the world. So in other words, in other scientists, other people say, Africa is the cradle of humankind. This is where humankind started. Which Homo species, according to this theory, which theory? Out of Africa hypothesis, was the first to leave Africa about 1.8 million years ago? And that is Homo erectus. And that is how Homo erectus ties up to the out of Africa hypothesis. It is said that Homo erectus was the first to leave because of bipedalism, and because of its bigger size, it was easier for it to traverse from one area to the other areas. The next question says, why is Africa regarded to as the cradle of mankind? In other words, what gives rise to this theory that says out of Africa hypothesis, or this hypothesis, sorry, out of Africa hypothesis? What evidence do scientists use to claim that Africa is the cradle of mankind? This is what they want to know. The oldest fossil hominids were found in Africa. Most fossils of hominids were found in Africa. We can add to that also. And they use genetic evidence. I'm not going into the genetic evidence now because the next question talks about it just now. Good? Please don't forget to post your questions if you have some questions regarding out of Africa, human evolution, or evolution in general for now. Let's restrict our questions to that section and we will deal with it as well. And as we go to the next section, you can post those questions and so on as we move along. Why DNA is carried by males only and can only be inherited from their male ancestors? Explain the statement. What does the statement mean? Simple. It means that the Y DNA is found on the Y chromosome. The Y DNA is found on the Y chromosome. And you know from your genetics that males only have Y chromosomes. So obvious from that then, they can only inherit this from their fathers. So it's the father that gives the Y chromosome that makes the child male. Okay? Without that Y chromosome, the child would have two X chromosomes, one from the father, one from the mother, and that would not be a boy, it would be a girl. So that's where we're talking about that. And the next one. Now here, this is a nice question in a sense that it's linking meiosis that you've learned with evolution. They're saying, explain the role of polyploidy in terms of evolution. How does polyploidy come up here? Interesting. First of all, you've got to explain what it means. When homologous chromosomes do not separate during meiosis, certain gametes may have pairs of chromosomes. Instead of being haploid, they are diploid, resulting in triploid or tetraploid zygotes forming. In other words, if a diploid gamete fuses with a haploid gamete, 2 plus 1 is 3. So that's triploid. If a diploid and a diploid, 2 plus 2 is 4, tetraploid. And you can get octaploid. So all those above 3, we talk about polyploidy. This may lead to genetic variation. Because of this, obviously if the chromosome number is changing, then the organism is changing. The genes that this organism has is changing. And this could lead to the formation of new species over time. And the next question says, tabulate 3 differences between natural selection, natural selection and artificial selection. We want to know the differences between them. Remember, they said tabulate. Look at the mark allocation, 7. So tabulate, you need 1 for the table. So you must draw a table. And your headings are natural selection and artificial selection. First one, environment causes the change in natural selection, whereas in artificial selection, the humans cause that. Artificial, you know. Because it is artificial, we can say that. Secondly, selected characteristic increases the chances of survival of that organism. Because that organism, nature selected it according to its suitability. Therefore, it has advantages over others. Whereas when we choose, chances of survival not necessarily increase. We have chosen this organism not for its advantage for itself, but for its advantage for us. For example, we want horses to run fast. So we look and we select artificially. We're not talking of genetic modification. We're talking of artificial selection. So we select, so there's no guarantee that they're going to be better in their environment. And it's a slow process, natural selection, over several generations, whereas artificial selection can be done very much quicker than that. We can add to this that natural selection, there's no cost involved and no effort, whereas in artificial selection, there must be human intervention and it may have some costs. With that, Luni, time for a break, I think.
Guys, we are going to take a very short break. Remember, I do post the pictures that Aslam is going through together with the questions. So if you just go onto facebook.com/learnextra, you'll find everything there. We are going to take a break, but do remember to stay tuned because we have more straight after this. Welcome back, Mindsetters. We're still doing life sciences with Aslam. Make sure, guys, you ask us any questions you may have on Twitter and Facebook, and tell us which province you're in and when you're writing life sciences. I see Brilliant is writing on Wednesday, and Dombizod is also writing on Wednesday. So all the best to you guys. Hope you learn everything that you need to learn. Hope you study very hard and you revise with us as we're revising with Aslam. So Aslam, let's take it away.
Thanks, Luni. Okay, those guys that are writing on Wednesday, good luck to you. Those that are writing on Monday, good luck to you as well. And we want to hear from the other provinces as well. When you're writing in your province, whether you're writing a common exam or you're writing individual school exam for the trials. Tell us that also on the site so we can get a gauge or get an idea of what's happening in the country. Coming back to this natural selection and artificial selection. Now, they then end of last year, this was in a prep paper, and end of the last year, they then put this as an essay to show the differences between natural selection and artificial selection as the last part. But you needed to explain natural selection, explain artificial selection, and give an example. We'll come to that when we get there just now. Just something on artificial selection that lots of learners confuse. Artificial selection is not a process where we take out genes from the organism and we exchange them. It's not that. Artificial selection is where we choose individual organisms with certain desirable characteristics. That means something that we like about this animal. Pointed ears, for example, in a dog, or a muscular horse, whatever we like in that organism for our benefit. And we then find another organism that has similar characteristics, or we take two organisms that have two different good characteristics. The one may have strong muscles, the other one may have long legs, etc. So it's a bit taller. And we try to breed these, interbreed those organisms to get organisms with both these characteristics. Again, remember, it's up to chance because genetics is going to come into place. If these are heterozygous, we would not be guaranteed to get those desirable characteristics if they are heterozygous for that. But we'll talk about it under genetics as well. But just so that you know that artificial selection is not taking out genes and altering the genes. This is simply taking organisms and breeding them because they have good characteristics, and we want to propagate those good characteristics in the offspring. So keep that in mind. The next question deals with speciation, also from the Prep Gauteng last year. The Isthmus Panama is a narrow strip of land that joins North and South America. According to scientists, the land bridge formed approximately 3 million years ago. Snapping shrimps, genus Alpheus, can be found on either side of the land bridge. There's a land bridge, and on either side of this land bridge, we're finding these snapping shrimp. The two groups are phenotypically similar. However, when the males and females from either side of the land bridge were brought together, they snapped aggressively at each other and would not mate. So they are similar species or similar organisms. They share similar characteristics because they are phenotypically similar. But when we bring the males and females from either side together, they don't want to mate. They are now considered to be two different species. The minute they mention in the paper two different species, then obviously the question is about speciation. Explain how the differences between shrimp on either side. Explain is your verb. Explain how the difference between the shrimps on either side of the land bridge could have arisen. So if they are new species, we need to explain how they became new species. Now I want to show you, and when we go through this answer, and this is why we put these questions together. We've taken samples to show you that no matter what the example, but the steps that you have to follow, whether it's natural selection, whether it is speciation, the steps to follow is usually the same. For natural selection, and speciation for speciation. Okay. Always, we start with this sentence. There is variation in the species, in the population. Before the land bridge formed, probably, the shrimps were able to interbreed. When they were not separated, they were allowed to interbreed, or we could say gene flow took place, same thing, in that population. Now, when this bridge occurred and the populations were separated, by this land bridge or geographic barrier, the two populations could not interbreed because they were separated now. So they are apart from each other, so the land bridge is causing the separation. Therefore, it's a geographical isolation. And this geographical isolation will lead us to call this a particular type of speciation, and that is allopatric speciation. The populations could not interbreed, or notice this is a slash, this is an alternative to saying could not interbreed, or you can say there's no gene flow. It means the same thing. Within each of the two separated populations, there was variation. We spoke about that there as well. Each group underwent natural selection independently. Why? Because the environment on either side of the bridge was different. Now what happens? Because of that undergoing natural selection independently, they become genotypically genes, and phenotypically their outward appearance becomes different from each other. So now, when the males and females from either side of the land bridge are put together, even if we take away this barrier now, so originally they were together, interbreeding. For some reason they got separated, can't interbreed. And when they were on either side, they developed independently from each other, and there may be different conditions on either side. And because of that, certain genes may have been favorable on one side and may not have been favorable on the other side. So natural selection made them genotypically and phenotypically different. In this case, they're similar, but not exactly the same. So now, if we take away this bridge, we put them together. That's what the scientists have done. They put them together. There's no barrier. The barrier was preventing them from interbreeding. Now when we put them together, they don't want to interbreed. So they do not interbreed, or there's no gene flow. Why? Because of reproductive isolating mechanism. It could be that they're not attractive to each other. It could be that they do not recognize each other of the same species. They become so different that they do not recognize each other of the same species. Or other reproductive isolating, the mating season has changed, not in this particular case. Those are other reproductive isolating mechanisms. The mating season may have changed. The courtship behavior may have changed. You know, different organisms may have different courtship. So all these things may change, and because of that, now they do not mate. They are now two different species. So you look at the points again. We talk about interbreeding originally, then we're talking of a barrier, then we are talking about different environmental conditions, then we are talking about natural selection, then we are talking about genotype and phenotype, and then we're saying cannot interbreed again, even if they put together. And the last statement, why? Because they are two different species. So the example doesn't make a difference. The process you're going to follow will still be the same. But you must contextualize your answer. You have to put these points that we are talking about in a real example. You can't just talk about these points on their own without referring to the context that the question is in. So keep that in mind. Next question from 2012 NSC paper 2. This is from version 2, I think. Read the following passage and answer the questions that follow. Thousands of wildebeest were migrating across the Great Plains of Serengeti in Africa. Some of them were large, some small, some strong, and some weak. Lions followed the wildebeest, catching and eating the slowest ones. Water was scarce, and only the strongest were brave enough to stop to drink from the water holes. Nice little paragraph. They want to know from you, identify one phrase in this passage which describes variation among the wildebeest. And that was, some were large, some were small, some were strong, and some were weak. Any one of those, these sets. This is one set, some large, some small. Difference, variation. Some strong, some weak. That's another variation. Explain how Darwin's idea of evolution by natural selection can be applied to this passage. So they've given you a case study there, and they want you to apply Darwin's theory of natural selection to this particular passage. Okay? You need to explain that. And there we go. First of all, there is variation in the wildebeest population. This is a compulsory mark. You must say this. There is variation. Whether you're talking about speciation, whether you're talking about natural selection, you must talk about variation. That these wildebeest have some differences among them. Some were maybe slow and weak. Those that were slow and weak were eaten by the lions. And they do not, because they're eaten by the lions, they do not pass on their characteristics to their offspring. And on the other hand, if you didn't discuss this, you could have said some were large and some were strong. They will survive and pass on their favorable characteristics to the offspring. Remember, this was a shorter question, so you didn't have to go into all the details. However, if they're giving you a longer question on natural selection, you're going to say there was variation in the wildebeest population and there was competition between them. You must mention that there's competition. Some had favorable characteristics, large and strong, while others had unfavorable characteristics, slow and weak. Those that had the favorable characteristics, strong and large, will survive. And if they survive, they can reproduce. And if they reproduce, they can pass this favorable characteristic to their offspring. They don't pass the characteristic as a characteristic, but as a gene. A favorable gene is passed. They have this favorable characteristic, or rather this gene, that makes them stronger and larger. And that is passed on to the offspring, and if this process continues from generation to generation, then the characteristic of the population will change. Now what we mean by that, that the frequency, just going to take this down, the frequency of strong and large will increase, and the frequency of weak and small will decrease. That means over several generations we'll see less and less of the weaker ones and more of the stronger ones. In this way the population evolves, not the individual, but the population evolves. All natural selection is telling us that certain genes, their frequency increases, the favorable ones, and certain genes, their frequency decreases, the unfavorable genes. Let's move on. One of the observations Darwin made during his study of pigeons was about artificial selection. In 1859, Darwin and Wallace jointly proposed that new species could develop by a process of natural selection. Using examples, describe natural and artificial selection and also highlight the differences between these two processes. Now, I'm not going to go through this entire answer because we've already looked at natural selection, explanation, and I've explained to you what artificial selection is. This is the essay question from last year's November 12, paper 1. This is the essay question, 17 and 3. What I want to do, more importantly, is to show you how you go about writing your essay. First of all, before you write your essay, you need to read. There's a whole passage here. One of the observations Darwin made and what have you is information, is additional info. Now what happens? They say using examples, describe. That same thing I'm talking about all the time. The verb. The verb says describe. What must you describe? Whatever comes after the verb. natural and artificial selection. You need to describe them and you must give examples. What many learners did last year, they concentrated on the example and they went to explain the example without explaining the processes, natural and artificial selection, and they lost a little bit of marks. And lastly, they want you to highlight the differences. So if you had to answer this question, then you would start by explaining natural selection. You're giving an example, the finches. There's 1 mark for the example, and then you're going to explain that. You could also use your example in the explanation. Then you would go to artificial selection and you would give your example, and you're getting a mark for that, and you would then explain that example. Lastly, you would highlight the differences between natural and artificial selection, which we have done already, but we're just going to put it up for you again. Those are the differences. Remember, in your essay, you will not draw it in a table. The table is just there so to show you the highlight, just to make you aware of it. And if we were marking, it would be easy for us to mark if this was there in front of us. But you will explain it in an essay form. Luli.
All right. Guys, remember to ask us as many questions as you can on Facebook. I'd just like to tell Aslam that Rufilo is writing a provincial exam on Monday. So all the best to you.
Okay.
We'll take a short break, guys, and we'll see you straight after this. Welcome back, Mindsetters. We're still doing revision for life sciences. All the best to everyone who's writing next week. I know it's quite hectic, life sciences. I never did it, but it seems hectic. Remember to go on to Curia, guys, and answer all the questions. Remember, you can only submit your answers after you've answered all 5 questions. You can't just submit without completing the questions, guys. And then it will tell you if the questions are right or wrong. Then if they are wrong, please just make sure you go through the questions properly and you make sure you answer it correctly. And then Aslam will go through everything right at the end of the show. I'd just like to remind you that we are playing revision shows as well for Maths Lit, Business Studies, and Geography. So if you watch throughout the day, Learn Extra will play those things for you guys. So Aslam,
thank you, Luni. We had one question. The learner wanted to know, first of all, whether we can write the essay in point form. Yes. Notice this is a memo. This is taken from a memo. You notice that it is written out, not in point form, it's complete sentences, but at least it's in a, you do not have to go into the type of essay that you write in life science. It's not the same as the type of essay you write for creative writing in language, or a history debate or a big essay in history or business studies. In life science, we are concerned more with the facts. You notice that 17 marks are given for the fact and 3 for synthesis. The synthesis mark, the 3 marks, is not a heading. Do not write synthesis and then write something. No. That synthesis mark, the marker looks at your essay and he gives you 3 marks, 2 marks, 1 mark or 0 for that. Obviously, if you have attempted the answer and you have some facts, you can't get 0. You must get at least 1. You must have some. But if you attempted the answer and we're asking about this natural selection and artificial selection, and you talking about speciation, then it's irrelevant what you're talking about, so there you will get 0. But if you present the facts without gaps, in a logical sequence, like the question told you, natural selection, then artificial selection, and then the difference, put that first, go in that order, explain natural selection, go all the way and do that, then artificial selection, do all of that, and then highlight the differences. In that way, you get full marks because you are following a logical flow in your answer as well. So make sure that you can, when you're giving points now to answer that other question, your point must mean something. You can't just put there a variation on its own. What is variation? You must be able to say that the organisms show variation. So we want, a sentence that can stand on its own, even if it's brief, but it must be able to stand on its own. But this is how the memo is given, and this is how we mark at the end of the year, so you can gauge by that. And in your lifetime, in our time, the memos were a trade secret. We couldn't have the memos. In your lifetime, as you're growing up, you have access to memo, so you can see how the answer must be set out. Sianda, ne? Sianda CC.
Yes.
Sianda CC, this is a shout out to you because you say you love life science, love science.
Yes.
Belinda, you heard that one. Love science. Okay, there we go. That's the essay. The other question was about modified genetic modification
What
and
is the
artificial.
difference from genetic?
Genetic modification, guys, is when we alter the genes. In other words, we isolate a favorable gene in an organism, and we take that gene out and we put it in another organism to make that organism better. For example, even genetic modification we use when we are manufacturing insulin using bacteria, where we isolate the gene for producing insulin, and we allow that gene to multiply in bacteria, and then we make insulin, and people can use that insulin as synthetic insulin for themselves as well. In that way, we are modifying the organisms, right? So understand the difference. We move on. The graph below shows the results of an experiment done to investigate the average time that fruit flies can survive without food. Important. What are we investigating? The average time that fruit flies can survive without food. Starvation resistance. The average starvation resistance time is when 80% of the population has died out. The researcher placed 5,000 fruit flies offspring from the same generation in a large container without food. No food. The average starvation resistant time was recorded. The eggs from the container were collected. In other words, while they were starving and the rest of it, they didn't still stay away from reproduction. They had eggs that came out from there. And those eggs were collected, transferred to a new container, and allowed to hatch second generation. And the average starvation resistant time was recorded again. And the procedure was repeated until the 8th generation. So this procedure, they put the flies there, they took the eggs, they put them in another container, they didn't give them food, and so this process continued. And let's see what happens. They gave a graph, and they're telling you this is what happened. This is the average starvation resistance time over 8 generations. That's the heading of the graph. I'm doing that because you must know how to do that when you are drawing the graph. The x-axis, the generations, and on the y-axis, the average starvation resistance time, the dependent variable, the independent variable, the caption. And notice that our intervals are equal there all the time. In terms of a bar graph, your width of your bars and the distance between the two bars must be the same throughout. And in terms of year, you must have the correct interval. Identify the dependent variable in this investigation. That's easy, the starvation resistance, because starvation resistance time is dependent on the number of generations there. State 2 factors that should have been kept constant in this investigation. First of all, the temperature in the containers must be the same, because if we alter the temperature, then we're changing. We're not checking that. What are we testing for? We are testing for how long they can survive without food. If we change anything else, then we are not sure that the factor that we are changing for is indeed the reason for the change. So therefore, when we talk of constant variables, it refers to all other variables that can affect the experiment apart from the one you're testing for. So flies from the same generation or age must be used all the time. Flies of the same species we've got to use. Same amount or no, well, in this case we're not giving food, so all are not giving food, or the same, or the type of food and the amount of food must be the same. And the number of eggs used every time that we are changing this from one generation to the next. So those are some of the things that we need to keep into constant rather. Now, we've done this experiment. Now they want you now to explain this in terms of natural selection. And first of all, we must know that, and as we said, there is variety in the fruit flies population. There must be a variety in the fruit fly population. Some fruit flies would have, and now you're explaining that difference. Some fruit flies had longer starvation resistant time and others not. When there was no food, when there was no food, the fruit flies with shorter starvation resistance did not survive. They died. And the fruit flies with longer starvation resistant time survive. What are we talking about here? We first of all talking about competition Okay, let me just undo that. Undo. We're talking about whatever. Oh, okay, right. Competition. Okay, there's competition taking place because they are competing for space and food, etc. And we are talking about favorable and unfavorable characteristics. Can you see the bullets? The variation first. Then we're talking about competition. Then we're talking about favorable and unfavorable. Then we're saying survival of the fittest. Because those that were longer, they survived. And reproduced offspring with longer resistance time. Now what has happened? Genes or the genotype for the favorable trait were passed on because they could reproduce. If they survived, they could reproduce. And if they reproduced, then the genes, the favorable genes that they had, they passed on to the offspring. And therefore, over several generations, there would be more and more that can survive for longer, and less and less and less and less and less of those that couldn't survive without food for long. And therefore, over time, we will have more of these, and this is natural selection. Notice again, I keep going back to the fact that you've got to go back to what you're saying initially in terms of those 5 or 6 bullets. A question now says, describe how each of the following contributes to genotypic variation within species. Meiosis, mutation, sexual reproduction. Guys, you've learned this, not in evolution, you've already learned that in meiosis, you've learned this in DNA and in meiosis, and in DNA replication and the rest of it. You've learned about meiosis, you've learned about mutation. And in your paper too, you'll learn about sexual reproduction as well. You don't have to learn it twice and 3 times. You're going to use the same information from there and bring it in to see how it fits in with evolution. And what is it that's bringing here? We are talking about this big word. Because evolution keeps talking about variety, variation. So how did meiosis bring about variety? And that's easy. And in this paper, we are revising both at the same time, isn't it? We're talking about meiosis and natural selection, not natural, evolution. Good. So, first of all, in meiosis, during prophase, crossing over takes place. And genetic, now they're explaining what crossing over is. What is crossing over? Genetic material is exchanged between homologous chromosomes. Or we say that recombination of genes occurs of the whole story there. And they become different from each other. So this brings about variety. 1. Then during metaphase of meiosis, homologous chromosomes arrange themselves randomly. So we're talking about the random assortment. That means there's no fixed way in which these chromosomes are arranged, and there's no guarantee which chromosome is going to go to which sperm or which egg. And this brings about variety as well. So that's the first part, meiosis. So meiosis, there's 2 things. One is crossing over, and the other one is a random assortment during metaphase. Those are the 2 things that you talk about in meiosis. Then you go to the next one about a mutation, and all you're doing in mutation, you're explaining what a mutation is. It's a sudden random change or a change that occurs in the genetic code or in the gene or in the DNA. Any one of that would be correct. The last one, how does reproduction, sexual reproduction? What's the question? Sexual reproduction. First of all, a large number of gametes are produced during sexual reproduction. And the gametes are different because they are produced by meiosis. And now, the more important part here, different from the rest. There's a random fusion of gametes. There's no guarantee which sperm is going to fuse with which egg. Whether it's a cow, whether it's a sheep, whether it's a human being. You cannot guarantee that sperm 1 that leaves the body is going to fuse with an egg. There's no guarantee. In humans, for example, there's 200 million sperms during ejaculation. Only one of those sperms can fertilize the egg. There's no guarantee which one. So that also gives you, there's a randomness in that, and that randomness brings about variation. Also, because a sexual reproduction involves 2 different organisms. And if it involves two different organisms, there's an exchange of gametes taking place. And by virtue of the fact that there's an exchange of gametes taking place, obviously then the offspring would have some variety, because the offspring will not look exactly as like the father or the mother. It would be a blend between the two, depending on the dominant and recessive characteristics found in those two individuals. And that's how you would answer that particular question. Next question, now we're moving away from evolution towards DNA. And I'd like to start with this one here. The DNA fingerprints below were used as evidence in a court case in order to convict a crime suspect. Fraction of a DNA fingerprint was derived from dry blood that was found on the victim's belt with which he was strangled. Study the DNA fingerprints and answer the questions that follow. Okay, so they're giving you there suspect 1, suspect 2, and this is the evidence. Question, which suspect is most probably the murderer? Suspect 2 is the answer. Now, obviously, they're going to ask you why you say so. And why do you say so? Because the DNA matches that of the evidence. Here's the evidence. Here's the evidence. Here's that suspect. Look at the blocks. There. Not just matches, but it's identical to the evidence. Is there any way in which the suspect can prove his innocence? Yes, he can argue that the dry blood came from the victim himself or herself, that the dry blood was there and he used his blood and so on. And there's other ways that they can obviously talk about that. In which way do you think the forensic team can prove this claim wrong? Forensic team should also have made a DNA fingerprint of the victim in order to compare it with the evidence. And the last question says, if one of the suspects refused to give his DNA for testing, what should he be forced to do so? Again, you could say yes there, and you can answer that, your argument. But you must agree first and then give your answers. For example, he claims he's innocent, or it's his constitutional right not to give his blood, etc., etc., etc. You can read that. Or you can say no, we can say no, because his right to privacy cannot be violated, he cannot be forced to do anything against his will, and so on. The first one, you're saying yes, he must be forced because he must be able to prove his innocence or guilty, and we need to solve this crime. On the other one, we're saying no, he doesn't have to be forced because of his constitutional rights and so on and what have you. Lastly, why do investigators take a sample of the victim's DNA at the crime scene? This is what would answer the top question as well. To check whether evidence found on the scene doesn't belong to the victim. Because if that belongs to the victim, we're going to be looking for DNA for somebody that we buried already. The victim will be gone. So we must take that. So we have that. We must have the suspects, and we must compare this evidence against all of those things to come to a conclusion. Luni?
Right. There's just a quick question here from Dombizoto. She's asking, so genetic modification and cloning, are they similar? If not, what's the difference?
We'll take that after the break.
All right. So, Ndomzara, make sure you watch straight after the break. We will answer your question. Guys, we will be back straight after this, so make sure you stay tuned. Welcome back, Mindsetters, from that very short break. Remember, guys, we do have Curio going on for life sciences as well, so you just need to answer those 5 questions, and Aslam will go through the questions with you later on during the show. But right now, Ndombizoto Aslam asked, so genetic modification and cloning, are they similar? If not, what's the difference?
Genetic modification and cloning. Cloning is a form of genetic modification. Cloning comes under the section of genetic modification. Why? Because we are modifying an organism. We're interfering with the genes. We're taking out stuff from one organism, and then we're adding the egg of another organism into that, and so on and so on. So it is genetic modification.
All right.
Anything else we got there? No?
Oh, yes. I don't know this boy's name, but it's Rifil or something like that, is asking, must we always write the subheadings for essays?
Yes, it is easy for yourself, for a start, to write the subheadings. And sometimes if the headings are not given in the question and you need to select those headings, there's marks even given for that. But it is nice to give your essay a structure that you know what you are doing. So you finish one heading and you move on to the next. it will help you. You know, there's no penalty for that, and it's going to take you a minute to write that, not even a minute, a couple of seconds. So keep in that habit. It makes it easy. It gives you structure in what you're doing as well. Coming back to this question here on DNA fingerprinting, remember this type of a question, the way they ask it is almost always the same. They would give you strips there, your barcodes, and with the evidence and the suspects and the victim sometimes. And this question didn't give you the victim, and they asked you a question about the victim. So in terms of that, you need to know. First of all, it's a matching exercise. You've got to match the evidence with one of the suspects. And when you're doing paternity testing, that's slightly different because the child will not have exactly the same bars as the father. In that case, we look at the one that has the more commonalities, the one that has more frequent bands that are similar, and that one is most probably the father. Because you can't have an identical, because you as a child, your barcode or your DNA fingerprint is different from each of your parents, and it may have similarities to both of them. So be careful about that. Question 9. Now we're moving to a genetic question. Study the genotypes and phenotypes below that shows how the alleles for color blindness are inherited. X and Y represent the sex chromosomes. Remember, they don't always have to tell you that X and Y represent. The minute they tell you it's a sex-linked disease, and in this case it's color blindness, you're supposed to know that color blindness and hemophilia are sex-linked diseases or disorders. So you need to know that. The minute we talk about sex-linked disorders, you must use the letters X and Y in your answer. You must use them in your answer. But here they've given it to you. So they've given you the genotypes, and I'm sure some of my friends, if they're watching, they're going to be smiling because I used their names here in this question. So Hamida here is XB, normal female. Farida, on the other hand, she's XB, X small b. She's a carrier female for color blindness. Ramona, XB, she's a color blind female. MV, on the other hand, X capital BY, he's normal. And Hagi, I'm hoping he's watching as well, X small b Y is a color blind male. Okay? So they've given you the genotypes and the phenotype of all those individuals. They want to know why color blindness is referred to as sex-linked disorder. That's a famous question when it comes to sex-linked disorders. They want to know why is this particular trait known as a sex-linked disorder, and the answer is always the same, because the genes for this particular disorder, in this case color blindness, is carried on the sex chromosome. It's on the same taxi. Don't write that in an exam, guys. The taxi is not, there's no marks for that in an exam. But what I'm saying is, if the chromosome is considered to be a taxi, then this colorblind gene is a passenger on that taxi. So wherever that chromosome is going to go, that passenger is going to go with it. That's what we mean by a sex-linked gene. That means the way maleness is determined and femaleness is determined, then the gene is on that same chromosome. So that is why it's there. Is color blindness caused by a dominant or recessive allele? Explain your answer. Let's go back. Look at the letters that we use. XB, normal female. XB, small, color blind. So the minute we use a capital or an uppercase letter, it indicates in genetics dominance. And when we use a lowercase letter, we indicate recessiveness. So they want to know, is it caused by dominant or recessive? From our table, it has to be recessive. Explain your answer above. The trait only shows up if it is in the homozygous recessive characteristic, not in the heterozygous. Let's go back to the table. Homozygous dominant, not colorblind. Heterozygous, not colorblind. Homozygous recessive, colorblind. This one here, remember in a male, there's no homozygous and heterozygous because the Y chromosome doesn't have something to counteract this one. So there's only one. So you can't talk of heterozygous or homozygous, but only when a recessive one is there, then the person is colorblind. Explain why Hagi is colorblind. Why we ask that question? You may say, oh, what the hell's wrong with this guy? Why did he put this question up when he has the recessive trait? But notice, Farida, she also has the recessive trait, but she's not colorblind. She's normal, but a carrier. Now, why is Hagi? He's got a small b here, but he's colorblind. And the reason is because Hagi has only one X chromosome, and this colorblind gene is carried on the X chromosome. It's not carried on the Y chromosome. They say the Y chromosome is too small to carry too many genes, so therefore very few genes are carried on the Y chromosome. So in his case, there is nothing else to counteract that small b that he has on the X chromosome. The Y chromosome is not going to help in this regard. So the minute he has even one small b next to the X chromosome there, he would be colorblind. Ramona and MV marry. Ramona and MV marry and have 2 children, a son and a daughter. Okay, a son and a daughter. Represent this genetic cross by using the information in the table above in order to determine the genotypes and phenotypes of the son and daughter. So in other words, what have they asked you to do? They've asked you to make a genetic cross using a diagram. Whether it was this one here or it could be anything else, the format of this is always important. And let's look at this one. So we say P1 or parent, phenotype, MV was normal from the table, Ramona was colorblind from the table. So obviously then MV's genotype would be X capital B Y, and Ramona's genotype would be X small b from the table anyway. You got that from the table. What do you do next? You write meiosis. We normally write it here, because meiosis takes place to separate the gametes. In this case here, it's XB, capital B, and Y. And in her case, they're both the same. Or you could have even written X small b there. But because it's the same, we wrote it once only. We prefer to use Punnett squares. So meiosis takes place, and then we write fertilization there. Whatever is on the left, keep it on the left. This is not a rule, it is my rule to make it easy for you so you don't confuse yourself. Whatever is on the left, keep it on the left. Whatever is on the right, keep it on the right or above. So there's MV's genotype or his gametes, and here's Ramona's gametes. And what does a Punnett square do really? A Punnett square is just there to show you if any one of these gametes fuses with any of those gametes, what are the probabilities? It doesn't tell you that this is the first one, that's the second one, that's the third one, that's the fourth one. It means every time fertilization takes place, it could be any of these. That's what you need to understand. That it's a case of chance, probability. We are predicting what can happen. Okay? What happens there? XB and XB there, XB small b, heterozygous. That one, capital again, that one, heterozygous. Now this is a Y. The Y doesn't have a B, but that's XB small and Y. And that one is XB small and Y. So now what happens? The offspring, the females are both heterozygous. So they are carriers and not sufferers. But both the males are colorblind. So in their case, they're going to have phenotype normal daughter and colorblind son. Now, why do we say normal here? We prefer to leave out, when we're talking about phenotype, we prefer to leave out the word carrier, because in a phenotype, if you look at somebody, if you look at Looney or you look at my eyes, can you tell whether I am colorblind or not? You can't tell that from just looking at my eyes. So my phenotype is normal. I have the genotype that I'm carrying. I'm carrying the trait for colorblindness. I'm not suffering from colorblindness. And I can't be a carrier of, I've carried the gene, not the phenotype. Okay? So notice the marks given here. You're getting a mark for if you write P1 and F1. You're getting a mark for meiosis and fertilization, obviously in their right places. So you've got a 1 mark there, 1 mark there. You would have got this right if you wrote the right phenotype and the right genotype. The whole thing must be correct. You would have got right 1 mark for all the correct gametes, either in the table on the Punnett square or if you wrote them out there. And you would have got a mark for all of these things being correct, and finally answering the question here at the bottom. There's the marks that I've given you already here, there, and there. in terms of that. Now, whatever genetic problem you do, you must set it out this way. When the disorder, if it's a disorder like albinism, is not a sex-linked disorder, then you do not use X and Y. You do not use X and Y. You use ordinary capital letters and small letters, like a normal genetic cross. So please make sure that you keep that in mind. Good. That was a diagram. So it did three things for us. One is showed us how to set up our genetic problem. Secondly, it told us when it's a disorder, how we answer. We must use X and Y. And while we did that, we discussed that if it's not a disorder, but a sex-linked disorder. If it's not a sex-linked disorder, then we do not use X and Y. This one now we're going to the other format in genetics, and that's the pedigree diagram. Study the pedigree diagram below showing inheritance of black or brown spots in Dalmatian dogs. The color of the spots is determined by a gene which has two alleles. The allele for black is dominant over brown. Now, in a pedigree diagram like this, the first thing you do, you try and put the genotypes without answering the questions. Now, what does it tell you? It tells you that brown is recessive. Now, here's the key. They tell you here, the ones that are clear are the ones with brown spots. The ones that are shaded are the ones with black spots. So, if an organism must have brown spots. Our old law on genetics, when the cat is away, the mice can play. That means the recessive gene will only show itself if it is in the homozygous. There must be no cat there. What we mean by cat? The dominant one. So if we go to our diagram here, we can then go to all these ones that are clear, and we can write in the genotypes, because they can only be that. This is what you must do when you're doing a genetic pedigree diagram. Now, if, for example, this was a sex-linked one, then you will obviously not use letters on their own. You'll use X and Y. Go to all the males and put XY. Go to all the females and put XX first. Then go in and put the dominant and recessive characters. In that way, you're going to be understanding it easier. Okay, now let's answer the questions. So we've done that. But by the way, when we do this, what can we determine? This child's got two small b's. That means this parent must have had the small b, and this parent must have the small b. But they are black, so they must have a capital B. And that's how you work backwards and forwards. I'm not going to do the whole diagram. I'm going to answer the questions, just to show you how to do it. So first fill in all the recessive ones, because they have to be homozygous. Then go back to their parents or go down to their children. If they are homozygous, they must give one to each of their children. And in that way, you work your way up or down. Now they want to know, name the color and gender of each of the following. B. B in the diagram is a female. B, female circle, plus that comes from the diagram here, and it's black. And G is a male, brown. G is a male and is brown. The key gives you that. Name the genotype of A, F, and E. A, we've given it to you there. A would be heterozygous. F, now here's F. Let's look at F. Here's a parent of F, that one there and that one there. This is already small b, isn't it? The mother has the small b's only. So F must have the small b. But what do we see here? F is black, and black is dominant. So F must have the capital letter. So we just write it like that. That's F. And the last one was E. E on the side. The E was done already for us because it's clear at the homozygous. Now, if C and D have another puppy, what is the probability of each of the following? The puppy being female. Remember, when any male and any female have children, offspring, the chances of them being male or female is always 50% over 50%, 50-50. Why? Because if we look at them on a Punnett square, X, that's the mother, X, Y, that's the father, XX, XY, XX, XY. So see, 50% male, 50% female. Whether you have 100 children or 150 children, the next one, the chances, genetic chances, is 50%, 50%. Last one there, of the puppy having black spots, is 75% because they are heterozygous, C and D. C and D are heterozygous. Am I right? Yep, they would be heterozygous. So therefore, there'll be 75% chance of them having puppies with black spots. With that, Luni.
All right, guys, we are going to take another short break. Remember that Curio is still up, guys, so you can answer all your questions. I see some of you are struggling with it, so I will post the instructions again on Facebook because the instructions are there, but I'll personally post them on the comment block there on Facebook. So do stay tuned, guys. We are going to take a very short break. We'll be back straight after this. Welcome back, Mindsetters. We are still doing life sciences with Aslam. Just a small question here from Hafsa. She's asking, I'd like to know the advantages of bipedal and hominids.
Hafsa, good question. She wants to know the advantages of bipedalism to hominids. Okay, that's for evolution. Remember, it's more for the evolution. Bipedalism, by the way, first of all, bipedal, standing on 2 feet, walking on 2 feet. So what is the advantage of us being upright? First of all, for females, they can use their hands to carry their babies, in humans, for example. Okay, and they can use their hands for other cause. They need to carry anything. They need to carry water. So in other words, if you are walking on your fours, you can't carry the water with your hands. Now that you're standing upright, your hands are free to do whatever you want to. Apart from that, you're going to lose me while I go down, because obviously the camera won't be there, and that's going to tell you if you can't see me there, then I also can't see that far. So when I go down and I'm on fours, my vision is limited to what I can see on the ground. But when I stand up like this, my vision is much wider. So you get a wider vision being bipedal. And the second one is that we can use our hands to carry other objects. Also the sexual dimorphism, that we are different males and females, but it's not an advantage. In other primates, in baboons and gorillas, the males actually use, Belinda, I'm hoping you're listening carefully, males use bipedalism to show off their genitals. Okay, males in humans shouldn't be doing the same. I don't know though. Okay, let's carry on with question 11. Adapted from 2011, question 1.4. Study the diagram below, which shows 3 generations of snapdragon plants, and answer the questions which follow. Use the following symbols for the contrasting alleles. Capital W for white, capital R for red. Now, the minute this comes in, your question, that is a capital letter for both the characteristics, that already tells you that this can either be incomplete dominance or codominance. Why? Because it shows that neither one are dominant over the other one, the minute they use the capital letter. So that's the first thing you must understand. All right, so they give you the whole diagram there, and they're giving you the clue, the key, snapdragons with pink flowers. That's another clue given in a question. The minute you see that, if we're taking red and white, for example, and we're getting another color coming from that, or any 2 characteristics, and we're getting a blend forming a new characteristics, that would be incomplete dominance. And if, when we have this situation and the offspring has both showing up in the offspring, for example, if we had red and white and we had a plant that had white with red spots or red with white spots, that means both are showing up in a phenotype, then it is codominance. This one is incomplete, okay? State the kind of dominance shown in the diagram above. I've given you that already. It's incomplete. Use the symbols R and W and write down the genotypes of each of the following snapdragon plants. So A was red, B was pink, and C was white. The one that is pink is the easiest one to do first. That was B, I think. Yeah, B is pink. That one will have both, isn't it? And if A was red, it will obviously be homozygous for red. And if C was white, it's going to be homozygous for white. Now, you can use the same question, and we change that thing and we make it now codominance. Everything else will change. The genotype won't change, but the phenotype, this is still going to be red, that is still going to be white, but this one will be red and white. But in incomplete, it's pink. It's a new color. Remember that. Okay, I was going to do this question, but I'm going to skip this one now for genetics because we've done quite a bit. Otherwise, we won't get to the others. Let's go to meiosis. I'm going to do one of the two that I have here and the essay. Study a diagram of a phase in meiosis below and answer the questions that follow. So they're giving you a diagram. First question says label parts B, C, and D. Before putting the answers, let's just do it on the diagram. It'll be easy for us. B is pointing to this little circle there, and that would be the centromere. And C would be a chromatid. Remember, it's pointing to one strand only. And D is the point of crossing over, and that is known as the chiasma. If there's more than one, chiasmata. Good. There's your answer there. Name the process in meiosis illustrated in the diagram above. What is happening there? Yeah? Tell me. Yep, it is crossing over. Now, what is the importance of this process? Crossing over, the only importance is exchange of genes allows for genetic variety. Draw a diagram of the structure labeled A to show its appearance after crossing over. And that's what you should have drawn. You had to show that part there. If you look at the diagram again, going up, notice up to there, this is what crossed over. That part of that chromosome came onto this one here. And they asked for A, this one here. They didn't ask for the whole thing, they only asked for A. So that part remained the same, that part remained the same, only the top part changed. And there you have it in your. So you need to, what is the examiner testing? He wants to understand whether you know what we mean by crossing over. And this diagram will teach you that. So notice that this chromosome, this is what it's looking like now, and before it was gray, all gray. Now it's gray and colorless in this particular case, or whatever color you're going to say, gray and white or whatever, well, gray and black in this diagram. Okay, so that should stick in your mind that when we exchange material, the chromosome is not what it was before, so it's different. And this difference brings about variety. So therefore, like for example, in our own families, our brothers and sisters, we may look very similar, but there are distinct differences between us. The next question, again, same thing, they got there, label C and D. In this case, C would be spindle fiber, and D would be, here you see, look at the curly bracket there. It's showing you both these chromosomes. So you had to label it as homologous chromosomes, not just chromosome. Okay, and there you have the answer there. Identify stage represented in the diagram above. They can't ask you phase anymore, so I change it to stage. What stage is this? You're not talking about a phase. Phase is metaphase, anaphase, and so on. Stages, is it stage 1 or stage 2? Which stage is this? Look at the chromosomes, they're lying next to each other. So therefore this is stage 1. Why? Because the homologous chromosomes are still lying next to each other. When they are single in metaphase and prophase, then that would be stage 2. And in anaphase, if they are separating and whole chromosomes, that means a chromosome with 2 strands is separating from each other, 2 of them, then that is stage 1. When you see single strands separating, the centromere has broken, that would be stage 2. Remember that when you are answering that. Name the process which results in part A being different. Again, from the last one, how does it occur? Homologous chromosomes cross over each other and parts of one are exchanged with parts of the other. State the importance, we've done that already. How many cells, this is an interesting question, how many cells will be formed at the end of the division of cells drawn in the diagram above? 2 cells. Because it was 1, it will become 2 in the first stage. Then they say how many cells or how many chromosomes will each daughter cell have when the cell drawn in the diagram above has completed meiosis. That means both stages. How many chromosomes do we have? 1, 2, 3, 4. And meiosis reduces chromosome to half, so therefore the resultant cell will have 2 chromosomes. Okay. This one here is the essay. We're not going to discuss the essay, just to put it up to show you how to set it up. Describe the mechanisms by which meiosis contributes to genetic variety. The verb is there. And describe how abnormal meiosis leads to Down syndrome and polyploidy. Also describe the advantages of polyploidy in nature. So, our answer then, we would describe first of all crossing over and random assortment, and then we will talk about Down syndrome, how it's formed, polyploidy and how it's formed, and finally we'll give the advantages of polyploidy in agriculture. I think I want to pause here to go to those curio, no?
Yes.
Let's go to the curio. And this is where we were with this particular question. The table shows the differences in amino. Let me go to the actual site to see what's happening there. If you look at the questions, only 38% got question 1 right, 23, 2, and 30 there, 26, 42 here. The biggest problem in 5 is many of you are just not answering the questions. But I want to look at this particular question here, and that's what we had on the board just now. The table shows the differences in amino acid sequence of the protein, albumin, and what have you. Please understand that when they're showing the differences, the less the difference they are, the more similar they are. So if there's less difference between the organisms, that means they are more similar to each other, and the more similar they are, then obviously they are more related, and that's what they recently separated. And your answer there would be D. It will be D, okay? So keep that in mind when you're answering that. We won't have time to go through the others, but make sure that you are keeping up with that. Any questions that you have, Luni? Nothing important there?
No.
Okay, I just want to then round up with what we were doing earlier. I just want to show you a question on protein synthesis. I have only 30 seconds to do that. And Belinda will put the pressure on. Okay, there we go. The question asks you to describe translation. That is how you describe translation. Transcription, copy, translation, making the protein, and you move from there. Remember, the messenger RNA, transfer RNA, DNA. DNA makes messenger, messenger gives the code for transfer RNA. And that is all, folks, I have to tell you this time. You need further, get to your books, go to the site. We've done all these sections before. I hand over to Luni. Thank you very much.
Mindsetters, thank you so much for tuning in. It's been great Saturday revision. Make sure to tune in next week Saturday as well, because there will be more revision. Stay tuned for more lessons, guys, throughout the day for Learn Extra. But we'll see you next time. Until then, goodbye.